So here we go.
So there are two types of cells, prokaryotes and eukaryotes. And we're going to use this chart right here to compare the similarities and differences between the two. So the first characteristic is which kingdoms of life include these organisms. So you want to tell us?
Sure. So our prokaryotes, we know that they're only bacterial species, and so there are only two kingdoms of life that contain bacteria. So it's going to be archaebacteria and eubacteria.
But if you can just remember bacteria, you should be good to go.
You should be good to go. And for our eukaryotes, it's everything else.
Right.
Everything else that has a nucleus, because that's the important thing that differentiates these two types of cells. All right, let's look at size. Prokaryotes?
Prokaryotes are bacteria, so they're very small.
Yep.
Whereas eukaryote cells tend to be larger.
Okay, so the easy thing is that if you know one side of this table, the other side of the table is just the opposite.
Right.
That's the nice thing about it. All right, let's talk about prokaryotes. Are they simple or complex?
They are simple. There's not a whole lot going on.
And then that means our eukaryotes are going to be?
Complex.
All right. And then whether or not they have a nucleus, we told you to remember no. So prokaryotes have no nucleus. And that means the eukaryotes, which is us, or you are a eukaryote, so it's going to have all the same characteristics that your cells have, and we know that your cells have a brain. Membrane-bound organelles.
So this goes right along with nucleus. You want to remember no. So all those fancy organelles like mitochondria, chloroplasts that have membranes around them, they are not going to be in a prokaryote, but they are going to be in a eukaryote.
Yeah, those guys are just too complex for prokaryotic cells. All right, let's go down and look at the second table. There are two main types of eukaryotic cells, so let's talk about what those two types are.
So the two types they're going to ask you about the most on the SOL are plant and animal cells. So once again, let's use this table to compare and contrast them. So starting off with nucleus. So these are both eukaryotes, so they are both going to have a nucleus.
Very nice. And mitochondria, well, we know that mitochondria gives our cells energy, and mitochondria has a membrane that surrounds it. So anything that contains membrane-bound organelles will have this particular organelle in general. So we know that plants and animals are both eukaryotic, so they're both going to have this particular organelle. Cell walls. Cell walls help to give the shapes that contain them the cells that contain them very specific shapes. So what shape is a plant cell?
So a plant cell is pretty square, pretty rigid, and that's because yes, it does have a cell wall. But animal cells, like your cells, do not have a cell wall, and so they tend to be more round.
Yeah, they tend to be more blob-like because there's nothing that's maintaining their shape.
Right.
Now, chloroplasts. Chloroplasts have a very specific job, Ms. Hines. Can you tell us what their specific job is?
Their specific job is to make food using the sun's energy, a process we've learned about this year called photosynthesis.
Now, we know that that's a defining process in plants. So plants have to contain chloroplasts. It also helps to explain why they're so green. And animal cells, us, we are obviously heterotrophs, so we cannot make our own food. Unfortunately, we don't do photosynthesis. A large central vacuole, that's sort of like the water tower of the cell. So what has this large space inside of it?
It's going to be something that needs that water for photosynthesis.
Yeah, so it's only going to be plants in this case. So animals do not have a large central vacuole. We do have a vacuole, but our vacuoles tend to be very small in size.
Okay, so now we're going to run through this list of the most important organelles and what they do. So starting with the nucleus. The nucleus is the control center of the cell. And that's because it has what molecule inside of it?
It has DNA. That's super important.
So let's write control center of the cell.
And it contains our DNA. And it's just something to remember from our last SOL review. DNA is always stuck in the nucleus. Why, Ms. Hines?
Because it's a really fat molecule. It's double-stranded.
All right, let's look at ribosomes. Ribosomes are kind of unique in that they're found in all cells, whether prokaryotic or eukaryotic, because they don't have a membrane. But they have one very specific job.
And that is to make proteins.
So they are the site of protein synthesis, which we also talked about in our last review. All right, mitochondria, mighty mitochondria. What does the mighty mitochondria do for us?
The mighty mitochondria makes energy, and in the cell, that energy is in the form of one very specific molecule.
3 letters, ATP.
ATP, energy.
And so, question, what process makes this ATP in the mitochondria?
Good question. That process is called aerobic cellular respiration.
So if we were to ask you, for instance, where does cellular respiration take place in a cell, you should be able to tell us mitochondria. Where does photosynthesis take place? You should be able to tell us
the chloroplast,
which is our
brings
next
us
organelle.
to the next line. Yeah, so we can write down that the chloroplast is where photosynthesis happens, and we can write that the chloroplast makes food for the cell.
And just so you guys know for sure, to be specific, the food that we're talking about is something called C6H12O6 or glucose.
Right.
All right, the cell membrane. All cells have a cell membrane. And what's the cell membrane's job?
The cell membrane is the door. It controls what enters and exits the cell.
So another way of thinking about it, we can say that the cell membrane basically maintains homeostasis.
Right.
So it maintains balance for the cell. And keep in mind, guys, the cell membrane is one of those crazy terms. It has 3 other terms that mean the same exact thing.
Yeah,
so if we
let's jot some
say
of those down.
the cell membrane, we're also talking about the plasma membrane. We're also talking about the fluid mosaic model, which is a really crazy term. And it's a long one, so we'll see if we can squeeze it in. And then there's one more based on 2 rows of a very specific molecule.
Which we're going to draw a picture of in a sec, and that's called a phospholipid bilayer, and we'll talk more about that.
And just remember, everything that we're writing on the screen, whether it's on a line or not, you need to make sure that you include it in your notes as well. It'll help you for the SOL. All right, let's look at the next one, cytoplasm.
So the cytoplasm is like the stuffing. It's just the fluid filling of the cell. It's mostly water.
Okay, and that's really all you need to know about that guy. The next one is the lysosome, and we've been talking about lysosomes all year. So what is a lysosome's job in a cell?
So a lysosome's job is to break down and sometimes recycle molecules that aren't needed anymore by the cell.
So it's kind of like the garbage can or the recycling center.
Yeah.
So it's pretty easy to remember. So again, garbage can or recycling center, that's the job of the lysosome. And then our last major organelle, and obviously this is not an all-inclusive list, but these are the most important guys that we want you to know for the SOL, is a vacuole. And we talked about a large central vacuole before, but we haven't talked about just in general what vacuoles do.
So vacuoles are kind of like closets. They're there for storage for the cell. They're used for storage. So in this next part, we're going to take a look at 3 different types of cells and just point out a few structures that you might be asked to identify. So first off, let's talk about what each of these cells is.
All right, if we look at this very first one in the upper left-hand corner, we see that this is bacteria. We know that it's bacteria because it has some funny-looking structures hanging off the back end. So we know it's bacteria, number 1, because it has this weird coating on it. And this weird coating has these things sticking off of it that sort of look like little tiny hairs.
Yeah.
And so we're going to call those cilia. And cilia can be used to help with attachment. They can also be used for movement. But really the main structure that's used for movement on this guy are these long whip-like structures that are coming off of the bottom end, and these guys are called flagella. And really the only cells in our body that have flagella, and it wouldn't be us, it would be the male version of our species, would be sperm.
Right. So just a couple other things to point out. Notice some of the organelles that this bacteria, this prokaryote does have. It does have a cell membrane.
Because all cells do.
Right. It does have ribosomes, those little dots to make proteins.
Because all cells do.
But then when we look at the center of this cell, we do not see a nucleus. Instead, we see a whole bunch of loose DNA in the center.
And remember, there's no nucleus. That should make sense to you, right, Miss Hines? Because it's a prokaryote, and we should remember no. All right, let's jump over to the other side. We have a very structure that should be not unfamiliar to you because it makes up who we are.
Right. This is an animal cell. And a couple of ways I know it's an animal cell, when I look in the middle, I see this one structure that really stands out. It's got DNA inside of it. This is the nucleus.
Also the control center.
So I know that this is a eukaryote because you do have a nucleus. I noticed that it's round. It does not have a cell wall. Instead, on the outside, there's just a cell membrane. I see some dots. Those are my ribosomes. One organelle I see that I did not see in my prokaryote are these guys right here. They look like ovals with little swirls inside.
And they're mighty.
Right. Those are my mitochondria.
And, you know, we often compare those things to, we say they look like kidney beans, or they're a bean-like structure to help you remember what it is. Is there anything else that we want to point out on that guy?
I don't think on here. Let's jump down to our plant cell.
Okay.
So how do we know this is a plant cell, Miss Cook?
Well, we know this is a plant cell because it has three things that animal cells never have.
Okay.
The first thing that we see, which is kind of large on the inside, it has a large central vacuole, so that's our water storage, because plants need a lot of water to do photosynthesis. And speaking of photosynthesis, they need another unique organelle that we don't have, which helps to give the plant its color, and it's the site of photosynthesis, and it's called the
chloroplast.
So we have chloroplasts here. Notice that these are not like mitochondria because every year people mix them up. Chloroplasts have these little stacks of pancakes, or we can say they look like stacks of cookies or hockey pucks that are found inside of them. And then another major difference would be the shape. So you'll notice that we have two barriers that are defining this particular structure. The inner barrier is the cell membrane, which is not unique because all cells have it. But the outer barrier is unique in that it's only found in plant cells, not found in animal cells, and that's our
cell wall.
And everything else is going to be about the same. So we have a nucleus because it's a eukaryotic organism. We still have mitochondria. That's important to know. Plants have both because they still need to be able to make ATP. And then we also have like the Golgi and the endoplasmic reticulum, both the smooth and the rough. But those are the major things that you need to know.
Let's go ahead and talk about photosynthesis and respiration. So photosynthesis uses energy from the sun to make chemical energy or food. And now we're going to go ahead in this space and we're going to write the chemical formula both in symbols and numbers and also in words.
All right. So what do we need to get this process started?
So we're going to need some water, some H2O. We know that all plants need it. They store it in their vacuole. We're going to need it for photosynthesis.
We also have to add a specific gas.
Right, and that gas is carbon dioxide. It's the one that we breathe out, and then plants take it in.
So that means that we have a relationship.
Right.
That's important.
And then to really get this thing going, we're going to need an energy input. And again, that energy comes from sun or light.
And then once we have those three things, we get our products for this reaction, which are
C6H12O6, which is
glucose,
our food. And then also, what is a waste product to the plant, we get O2, which is oxygen.
And that's really important because without that, there'd be no oxygen on the planet.
Right.
All right, let's jump down and look at number 5. Aerobic respiration breaks down
glucose or food
to make usable
energy.
And that's always going to be in the form of what?
ATP. So remember, ATP, energy.
All right, in the equation for respiration, we're going to do the exact same thing. We're going to give it to you in the chemical formula, and then we're going to give you the cheat sheet version on the bottom. So basically, we're just flipping the reaction around, right? We're undoing photosynthesis.
Exactly. So we ended with glucose and oxygen. Now we're going to take our food, our glucose, and our oxygen, and we're going to make CO2.
That's our byproduct.
Water.
Another byproduct.
And
most importantly,
ATP.
Which is the whole reason why we're doing this particular chemical reaction.
Right.
So notice there is a cyclical relationship between photosynthesis and cellular respiration. That's important. All right, let's jump down and look at number 6, seeing as how we can see it on the board here. There are 2 types of cellular respiration.
Aerobic and anaerobic.
And those words sound very familiar. So what's the difference between them?
So aerobic kind of sounds like air, right? And that's because aerobic respiration uses oxygen. You have to have oxygen for it to happen. And if you look back up at the equation we wrote, we have that in our equation. Here's our oxygen. Anaerobic, on the other hand, does not use oxygen. And you can remember that the N in anaerobic is for no. There is no oxygen.
And just so you guys know, sometimes on the SOL, instead of referring to it as anaerobic, it might also be referred to as fermentation, which means the same thing. And it could be lactic acid fermentation, which is that burning that you feel in your muscles when you start to run out of energy, or it could be alcohol fermentation, which is what's used to make things like wines and beers. All right, let's look at number 7. The cell membrane lets some things enter the cell but keeps other things out. It is
selectively permeable.
That's important. So now that we know,
So remember, the cell membrane is made up of phospholipids, and they look like this. They've got two tails and a head. So let's just talk about a phospholipid, and then we'll jump into the whole membrane. So let me label this phospholipid. Do you want to tell us about these two parts? They have very different properties.
yeah, the weird thing about phospholipids is that the top, the head, and the tails actually have opposite behaviors.
Okay.
So for the head actually loves water. And because it loves water, we know that it's what?
Polar.
Yep, so it's polar just like water. So it has a charge, is another way of saying that, so it can attract water. They like to hang out. If it loves water, we say that it's hydrophilic, so it's water loving. The tails are simply going to be the opposite. Instead of being polar, we're going to say that they're nonpolar. And instead of being hydrophilic, what word do we always use when we have a phobia of something?
We use that word phobia, so it's going to be hydrophobia. Hydrophobic.
So it hates water. So these two things, or these three things, the tails, the two tails and the head, even though they have a relationship with one another, they behave very differently. But those differences in behavior allow the cell membrane to do its job.
Right, so let's go ahead and draw how these things are arranged in an actual cell membrane. So in an actual cell membrane, the phospholipids exist in what we call a bilayer, which is two layers, and they're facing in opposite directions. The top layer has the tails facing down, and then the bottom layer we're going to draw the tails facing up. So the tails, the parts that hate water, are touching each other, and the parts that love water are facing the outside. So this is my phospholipid bilayer that we talked about before. But there's some other stuff in the membrane too that we should draw.
Yeah, but before we do that, there's something else that we need to pay attention to too. Because the tails are facing each other, they're creating a unique zone. What is that zone called, Miss Hines?
This is the hydrophobic zone.
Which makes sense because the tails hate water.
Right.
And then our two opposite sides, which are heads, are going to be our hydrophilic zone. So they actually love water. All right, let's go ahead and change colors for when we actually start to draw in the other two things that we need to talk about.
Okay.
All right, so let's choose blue. All right, so we know that we have phospholipids, or green, that's even better. So we know that we have phospholipids, but we also have some other things floating around in the membrane. Specifically, we have things called proteins. And these proteins are important because sometimes things cannot pass the membrane that desperately need to get from one side to the other. So the things that can't pass, we basically create a bridge for them, or a tunnel. And that tunnel is going to be created or using these transmembrane proteins, or these proteins that are embedded inside of the membrane. The other thing that we have honeycomb-looking structures that help to give the cell membrane its fluidability. And that fluidity, or the ability to change shape or to bend, is important. Otherwise, the cell membrane would crack. And what is this stuff called, Ms. Hines?
It's called cholesterol.
All right.
So cells constantly have to bring things in and out, and there's 4 different ways that the cells can bring in things it needs and get rid of waste that it doesn't need. And those 4 things are our 4 types of transport: diffusion, osmosis, facilitated diffusion, and active transport. So once again, let's use this chart to compare and contrast them. So the first thing we have to ask ourselves is this passive transport? Is it something that happens on its own without any energy, or is it active?
And just so you guys remember, all we're looking for, we're trying to see, are things moving from high to low, or are they moving from low to high, to figure out whether or not they require energy. So diffusion, will diffusion happen as long as there's a concentration gradient?
It will. So it's an example of passive transport.
Requires no energy whatsoever. So if I sprayed perfume in this part of the room, eventually it would make its way to the other part of the room, even without me giving it any energy, because things like to be equal, so they like to move from high to low.
So let's go ahead and label that diffusion happens from high to low.
Okay.
So in diffusion, what kinds of things are moving?
So we have different types of molecules. We're never talking about water here. That's the important thing. So diffusion is simply the movement of anything that's small enough to get across a membrane, right? So they tend to be small, but it's never water. That's the key. The next thing that asks, or that we look on the chart, asks, do we need any proteins for help? Well, the key is, look back to what's being transported. Is it small or is it large?
And here it's small, so we're not going to need any proteins. It can get across that phospholipid bilayer on its own.
Right, and then we've already answered this last question. Does it need energy? Well, it's passive, so the answer is
no.
Absolutely not. All right, let's look at number 2. Number 2 is osmosis.
And you can remember the O in osmosis for H2O, because that's osmosis is moving water.
All right, and osmosis is a specific form or a specific case of diffusion.
Mm-hmm.
So it's the diffusion of water. So we know that everything else, it's gonna be very similar to what we have for our first column. All right, facilitated diffusion still has the word diffusion in it, so we know that here's another special case of diffusion, so it's still going to be passive. Things are still moving from
high to low.
The only difference here is that we're not moving small molecules, we're moving large molecules. And because we're moving large molecules, we need a tunnel or a helper, and that helper is going to be our protein, so we do need a protein here. And then our last one?
Our last one is active transport. And again, obviously from its name, this is active. And the reason it's active is because molecules are having to move against the concentration gradient. They're having to get more concentrated from low to high, which they don't want to do. So the cell has to use some energy, some ATP, in order to get them to do that.
Yeah, so we're going to have to pump them or push them in a direction that they don't want to go normally.
And those pumps are
proteins. So we absolutely need proteins here. And what types of things are being transported?
It could be small, it could be large. It really doesn't matter on the size. The important thing is that it's moving from low to high, so we need energy.
Yeah, and the SOL is never going to ask you specifically what is being transported in active transport. They're just going to ask you, is this active transport? And the easiest thing to remember is, are we moving something uphill? Or are we letting things roll downhill naturally?
Water likes to go from areas of high to areas of low. Water likes to go from high to low. It rhymes. Concentration, just like other molecules. And the other important thing to know is that pure or distilled water, distilled means the same thing as pure, has a very high concentration. So let's draw a couple pictures here and see if we can think about where the water will go. So I'm going to go ahead on my paper and draw a cell, and you should be drawing this on your paper as well. And we know that cells have at least a little bit of solutes, of dissolved molecules inside them, like sugar and salt. So I'm going to put a few dots in there. Now let's pretend I took this cell and I put it into pure or distilled water. So this is a beaker with distilled water in it. You want to tell us what would happen?
Yeah, so there's one thing to keep in mind. Whenever we have water involved in any sort of scenario, water always wants to be equal. So anytime that we have a solute displaces water, meaning that there's less water present, regardless of what the space looks like. So if I'm looking at my cell, I know that I have some solute in there, which means that there's not as much room for water inside the cell. But outside the cell in my beaker, I don't have any solute, so there's plenty of water. So I know that my water concentration on the outside of my cell is very high. And on the inside of my cell, it's low because of the solute. Now, because water is low on the outside, or I'm sorry, high on the outside and low on the inside, and water always wants to be equal, which way is water going to flow?
It's going to go from high to low, so it's going to go into the cell. So let's think about what would happen to a cell if H2O goes into the cell.
So if something is entering the space is not going to get smaller, it's going to get
bigger. So the cell is going to expand.
And if the cell continues to expand,
it could even burst.
All right, let's try another example.
Okay, so again, I'm going to draw my cell, and I'm going to put a few solutes inside of it, a few dots. But this time, I'm going to take my cell, and I'm going to place it into very, very salty water. So I have a beaker here filled with very, very salty water. So I'm going to draw lots of dots to represent all of that solute that is outside of my cell.
So if I have lots of salt in my salt water, that means that I have less water. So I'm going to be low on the outside. Let's change the color so it's easier to see. So I'm going to be low on the outside, which means that by default I'm going to be high on the inside.
Right.
And again, water always wants to be equal, so in this case water is going to
flow out of the cell.
And if water flows out of the cell, is the cell going to expand or is it going to contract?
It's going to contract. It's going to get smaller or shrink.
Okay. Now there's one more scenario that we need to discuss. This other scenario involves
is when the concentration inside and outside the cell of solute and water are about equal. So if you take a look at this cell again, it has a little bit of solute on the inside. And outside my cell, there's also a little bit of solute. It's at about the same concentration as the inside. And I can tell that because the spacing between my solute is about the same inside and out. So there is no high and low. I can't put an H and an L. So what this means is a little bit of water might move in, a little bit of water might move out, but overall the amount of H2O will stay the same inside the cell.